What's up, everyone? So, today I'm going to go over some COVID-19 facts to educate more people on it. So, first, some symptoms. We have fever or chills, cough, shortness of breath or difficulty breathing, fatigue, muscle or body aches, and a sore throat and congestion. Some self-care treatments. So if you test positive, you're gonna want to stay at home, except to get medical care, and monitor your symptoms. If they worsen, call a doctor. Rest up and hydrate. Stay in one room and away from people so you don't expose anyone else to it. And if you have to go outside, mask up. If you test positive but you have a vaccine, you have to quarantine for 10 days, and if you test positive with a vaccine but you have no symptoms, you only have to quarantine for 5 days. Here's some myths and facts about COVID-19. Myth is the ingredients in COVID-19 vaccines are dangerous, and the fact is nearly all the ingredients in COVID-19 vaccines are also ingredients in many foods. Fats, sugars, and salts are some of them. One myth is that the natural immunity that I get from being sick with COVID-19 is better than the immunity I get from COVID-19 vaccination. The fact is, getting a COVID-19 vaccination is a safer and more dependable way to build immunity to COVID-19 than getting sick with COVID-19. Another myth is that COVID-19 vaccines contain microchips, and the fact is the COVID-19 vaccines do not contain microchips. Vaccines are developed to fight against disease and are not administered to track your movement. One more myth is receiving a COVID-19 vaccine can make you magnetic. The fact is receiving a COVID-19 vaccine will not make you magnetic, including at the site of the vaccination, which is usually in your arm. So here's some images of the rates, and there was Biden getting vaccinated right there. And here's a video.
It's one of the tiniest machines on the planet, about 100 times smaller than the average cell. It's so small that no scientist can spot it through a typical light microscope. Only with an electron microscope can we see its spiky surface. Despite its diminutive size, the novel coronavirus, dubbed SARS-CoV-2, has seemingly taken the world by surprise with its virulence. Cell biologists have been working to better understand the virus and the ways it differs from the coronaviruses that caused the previous epidemics. If we can reveal hidden targets and an Achilles heel, that could help us develop treatments to combat these viruses quickly and efficiently. To understand the unique nature of SARS-CoV-2, it's important to compare it to the other viruses in our world. A plentiful array of viruses surrounds us. In the ocean alone, there are an astounding number of bacterial viruses. In humans, scientists have identified several hundred species of viruses, not counting different strains, and it's likely that number is just the tip of the iceberg. Viruses are categorized by what their genome is made up of, either DNA or its closely related cousin, RNA. Their genome can be linear or circular, and some have fatty or lipid envelopes that surround and protect their genetic material. Despite being 1/100th the size of an average human cell, SARS-CoV-2 is considered a medium-sized virus. Coronaviruses use so-called fusion proteins that form spikes studding their surface, which give them their crowning name, to lock on to a host cell's outer membrane. While about 80% of common colds are caused by rhinoviruses and adenoviruses, the remainder are the result of coronaviruses. At Johns Hopkins University, researchers have used an avian model of the virus to focus on the unusual way coronaviruses assemble their progeny within a host cell. First, the virus enters the animal's body through a mucus-lined surface, the nose, mouth, or eyes. It uses its crown of proteins to fuse to a host cell surface, or a cellular package called an endosome, which is engulfed by cells. Once inside the host cell, the virus replicates its genome and uses the cell's machinery to make structural proteins critical to form new viruses that will soon escape the host cell. Instead of assembling by budding from the outer cell membrane like many viruses, coronaviruses assemble new viruses by budding in the early Golgi complex, a pancake-like structure that sorts and processes proteins and sends them to their final destinations, sort of like a post office. The viral proteins interact with each other, and the new genomes take a piece of the Golgi membrane as they form. They then move through the Golgi and into vesicles to be transported to the cell surface for release. Scientists recently found that coronaviruses are somehow able to neutralize the pH of the Golgi, which is somewhat acidic. It's thought to protect the spiky halo from being damaged by enzymes as the virus makes its slow way out of the cell in vesicles. These vesicles fuse with the host membrane and release the virus particles, which can then go on to infect new cells. It is our belief that continuing to pursue these lines of research could help identify new targets and strategies to interfere with infection and stop the virus in its tracks.